All right. So hi, I am Brianna, and it is my pleasure to introduce our seminar speaker this week, Dr. April Wright. Dr. Wright is an assistant professor of biological sciences at Southeastern Louisiana University, where she works on statistical phylogenetics, particularly integrating molecular and morphological information to answer cool evolutionary questions. Dr. Wright's also super passionate about teaching and incorporating computation in biology education, which I think is really cool. And apparently NSF does too, because Dr. Wright was just awarded the prestigious career grant, which is awesome. Thank you to everyone who is here and who attended the roundtable discussion on primarily undergraduate institutions before this. That was awesome. And graduate students, feel free to stick around for a half hour or so after a talk for a meet and greet and to ask any questions. And with that, please join me in welcoming Dr. April Wright.
Hi, everybody. Can you all hear me okay? Okay, cool. Let me get the slideshow up. Thank you so much for that introduction, Bree, and thank you for what's been a really fantastic day so far of interacting with, friends old and new. I know there's a lot of stuff that's sort of awkward and weird about seminar visits in our current situation, but this really has been excellent. You and the grad students have done a really wonderful job organizing all of this. So today, what I'm going to be talking about is some recently completed work and some ongoing work in my lab. And in particular, I'm going to be talking about divergence time estimation and trees that have been scaled to absolute, or as we sometimes call it, geological time. So on the left here in panel A, what you have is a tree that is not scaled to absolute time. So this is an undated phylogeny. Typically, when you see an undated phylogeny in the year 2021, you're going to have branch lengths that are in substitutions per site, or expected substitutions per site. On the right-hand side, we have a tree that is dated to absolute or geological time. So you can see on the bottom there is an actual time scale that shows millions of years of divergence on this tree. And going from one type of tree to the other, going from undated to dated is a little bit trickier than you might imagine. And in particular, over the past decade or so, the way in which we do divergence time estimation has really undergone a revolution. Just one second, I'm gonna minimize my panel of people who I can see there. So I've got a little more screen real estate. Okay, there we go. So divergence time estimation and the way that we do that sort of analysis has changed a lot over the past decade. And nowhere is that difference more obvious than in how we include fossil data. So data from extinct organisms, particularly morphological data from extinct organisms. So this is a figure some of you might have seen before. It's from a review by Tracy Heath and Brian Moore, and it was published a few years ago. What we have on this tree is bears and one wolf, right? So we have most of the crown bears represented here. And on this tree, we have two fossils. I've indicated them here in red. All right, so the way that we've historically sort of treated fossils in divergence time estimation is, we make the assumption that if we have a fossil, and let's say that fossil is 12 million years old, the group to which that fossil belongs, the clade to which that fossil belongs, must be at least 12 million years old. Right? That's a fairly straightforward sort of idea. The way in which we've typically incorporated this information in a divergence time estimation is what we refer to as node calibrations. So let's say, for example, this Cretziarctos beatrix, for example, that fossil is somewhere between 11 and 13 million years old. And that uncertainty, that value comes from the radioisotope dating of that specimen. Under a node calibration approach, what we typically do is we place a prior on the waiting time between when that fossil was discovered and when, or the age of that fossil, and when the split from which that fossil descends is. So for example, in that for Cretziarctos, we have a log normal calibration prior that says that probably that fossil is fairly close in age to the split that it subtends. On the other hand, we have this Zeroga scion down here. There's an exponential prior on that that says probably the age of the fossil is very close to the age of the split that from which it originates. Right? In practice, this sort of calibration is often fairly sensitive. So the ages that we estimate on a node are often very sensitive to the exact prior that we choose to kind of calibrate that waiting time. Something else that's a little bit unsatisfying about this type of analysis is that you can only use the oldest fossil in your group to calibrate a particular age. This also ignores character information. So when you're doing a node calibration approach, you say, okay, this fossil belongs to this particular clade. I know that without error. The age of the fossil is known without error. So we make a lot of very strong statements when we might have, a lovely morphological character matrix for that fossil. And so to a large extent, a lot of paleontologists were quite unhappy with this sort of modality of dating for quite a while. The neontologists also started to be fairly unhappy with this approach. You can have priors conflicting and interacting. So for example, that split that spawned Cretziarctos, if the age is actually down in that tail, we could specify an age for that split that is older than its ancestor split. So that doesn't make a huge amount of sense. Right? And so this framework has been sort of described as incoherent. This is a kind of famous quote from Heath, Stadler, and Holzenbeck 2014, this idea of node calibration as, this sort of Frankenstein approach to dating that is not terribly coherent either to the process and is sort of internally incoherent. Now, when I was in graduate school, there were some of the initial attempts at what we refer to as tip dating. In these tip dated analyses, we treat the fossils as true pieces of data. Okay. So something that's been true for many years is that people have been making joint molecular morphological phylogenetic trees. So this image that you see here is from one that I just sort of like from Lister et al. 2005. This was a joint molecular and morphological phylogeny of various deer. I've always liked it because it has the Mesopotamian fallow deer on it, which is a very cool looking organism. So in phylogenetics kind of writ large, people were starting to do analyses where we put our morphology in one partition, our molecular data in one or more other partitions, and we estimate a tree from both. But this hadn't really caught on in our node calibration divergence time estimations, where fossils are typically treated as calibrations rather than tips. And so in 2012, while I was a graduate student, Ronquist and Klopstein made sort of the first cohesive effort to really propose methods to deal with this. So you see on the left here, it's not important that you're able to read the tips, or on the right here, it's not important that you're able to read the tips. What they did was they did a study where they estimated a phylogeny of insects using a node dating or node calibration approach, but they also did what we would now refer to as, or what some people refer to as total evidence dating. So this is when you put your morphological matrix and your molecular matrix in the analysis and you do your divergence time estimation. And the way in which they did this was they developed a new tree prior, right? So tree priors describe the process of speciation, extinction, and sampling in both the present, so the extant sampling, and the sampling of extincts that leads to the observed phylogeny. So the tree prior is a prior on the actual sort of diversification process that generates our tree. So it's not on a specific node or a specific model parameter. Well, it's on the tree, which is a model parameter. But this is a prior on the tree itself. And so original attempts at this sort of analysis, at this sort of tip dating analysis, used what we refer to as a uniform tree prior. And that makes fairly few specific assumptions about the process of diversification that generated the tree. As the name suggests, uniform, we're not really saying any particular tree shape is more likely a priori than any other tree shape. And so this is a big step forward. Some follow-along studies, including some authored by myself, kind of showed that this uniform tree prior can have a little bit of an over-smoothing effect on some of the node ages. And so around about 2 years after that initial exploration, the first implementations of the fossilized birth-death process for divergence time estimation were made available by Tracy Heath, Tanya Stadler, and John Hulsenbeck. And I'm just going to run through really quickly what the fossilized birth-death model is. Right, so this is a mechanistic prior. Right, so we're specifying a process of diversification and sampling. Right, so this is a mechanistic prior on our tree. So this figure comes from a tutorial authored by a bunch of us, led by Joelle Barreto-Sotani, and this came out last year. Any of these, if you are looking at the references and you need to find a copy of the paper, let me know. All of these, I preprint everything, so there's no need to get stuck behind a paywall. So if you're looking for anything that I've cited in here, certainly let me know, and I'll get you a copy somehow. So I'm just going to step through the fossilized birth-death really quickly. Right, so in the middle is that that is the tree. So these processes, this is a graphical model. All of these parameters are pointing to the tree because these are the model parameters that define our tree. Right? So here indicated in gray, we have origin time, the time at which our process of diversification began in this group. Right here, lambda, this is the speciation rate. This is the rate at which lineages are added to our tree, to our population of possible lineages. Here we have mu, our extinction rate. And this is the rate at which lineages are removed from the population. So at which they go extinct, they can no longer speciate, they can no longer be sampled, they can no longer leave descendants. At the extinction rate or at the extinction event, they are done. Here we have our fossilization rate. You might also hear this referred to as the fossil recovery rate. This is the probability that a fossil is sampled. Right, so before the present, the probability that this is sampled. And finally, this is the sampling probability, the probability that an extant is sampled. You might notice that the sampling probability is in a square, whereas the other ones are in circles. What that indicates is that typically we know the sampling probability. This is often treated as a known value in a fossilized birth-death analysis, right? So if you're building a phylogeny of bears and you have sampled at least one member of every bear species for molecular data, you know that your sampling probability is 1, right? You sampled them all. On the other hand, I'll talk about ants later, and even this can be a little bit tricky, figuring out our extant sampling parameter. And what I want to point out is that different model parameters imply different tree shapes. So this is a figure from a manuscript by Rachel Warnock and I from 2020, was last year, I think. And what we have indicated in here is this is a 3-column figure. On the left-hand side is sort of the true tree, or the tree as it exists in reality. In the middle column is the tree that we are able to sample. And then in the rightmost column there, that is the model parameters that we're looking at. So for example, if we only have speciation in our tree, this is a pure birth process. We don't have extinction. We're not sampling extincts, right? We're not sampling extinct organisms. If we only have the speciation parameter, we should be able to get the tree mostly right. Right? We're not losing any lineages along the way, so we can mostly get it right. When we add extinction, right, we have some of our lineages not making it to the present. Unless we start sampling in the past, we will miss parts of the tree. Right? If we have tips that don't make it to the present, but we are not sampling in the past, we will miss that paleodiversity. Here we've added in extant sampling, right? So if we have speciation happening, there is extinction, so we're not sampling all of our lineages. And we're not sampling all of our lineages in the present. This is what that'll look like, right? We don't have all of our extants represented either. Now, when we add in fossil recovery, we start to be able to see some of those extinct branches that we were missing before. And so different parameterizations, different values for parameters will imply different possible tree shapes that we can reconstruct. A couple of things I just wanted to draw your attention to quickly. On that bottom tree there, you'll notice that there are some 2-degree nodes, so nodes that lie on a branch. So this model does imply that there is some possibility of sampling an ancestor and then sampling all of its descendants or some of its descendants. I know a lot of people who do vertebrate work, that possibility sits a little uneasily. This is something that people are much more comfortable with in invertebrate paleontology, which I'm going to come back to in a few minutes. And something that is really important, I think, is that we can simulate under this model. So we can develop sets of null expectations. And this is going to become important towards the end of my talk as well. So we can simulate under this model. We can take a look at what we would expect given a set of, starting conditions. Being able to simulate under your model and then estimate a tree and see if you get the same answer back, always sort of a primo thing to be able to do. All right, so this fossilized birth-death model is often implemented in what we refer to as a tripartite approach. So the fossilized birth-death is right here as the tree and tree model, right? So the fossilized birth-death is a model that describes the evolution of the tree. When we do divergence time estimation, that's not sufficient. We need to have models that describe other parts of our data. So for example, something that you might be familiar with is the idea of the substitution model. Right, so when you have your phylogenetic characters, let's say you have your A, C, T, and G, how do you have substitutions over time? How likely are you to go from an A to a C at a particular site in your matrix? Right, we have our clock model, right, which describes the rate of evolution, how rates of evolution are distributed across the tree and through time. And we're going to spend a little bit of time with each of those models today as well. Our data in this analysis are our phylogenetic characters. If you are a strictly paleo person, this might be just a matrix of morphological characters. If you're a neontologist, this might just be your molecular characters. If you, have a foot in each grave, this could be your molecular and morphological data. And kind of crucially, the fossil ages under this model are data as well. And this has some really interesting properties that we're going to talk about in a little while. And so the fossil ages being how old you think each fossil is. Usually you have a minimum possible age that your fossil could be and a maximum. Right, and all of this is typically implemented in a Bayesian framework. I know there are some likelihood-ish implementations of this model out there. And so this, typically works this way where we're looking to calculate, kind of a posterior probability of a particular solution given our data and model. And if you're really curious, we made this little graphical representation of our model where we have, our tree and our fossil ages and our tree model parameters, our clock model, our substitution model, and we're figuring out the posterior probability of that given our character information. And so that's roughly what this looks like. And again, this is from a review that I wrote with Rachel Warnick last year. So if you are interested in reading this, get in touch and I can get you a free to read copy. Now, what people often say to me at this point is, okay, I thought you said this was going to be more coherent than the node calibration. I don't know how to set priors on any of these things. Speciation rate, extinction rate, what am I supposed to do? And so I'm going to walk you through a case study that I worked on with Pete Wagner and Davey Wright last summer, I guess. Yeah, last summer, where we worked with synktons, right? And synktons are a temporally restricted group. So they're found in a fairly narrow time range. They're also geographically restricted. They're found in a fairly narrow range of geographic situations. These are Cambrian echinoderms. This is what a synkton looks like. It's like a tennis racket or like the first pancake off the griddle where you're like pouring batter and it's all over the place. So these are small marine organisms and they have a surprisingly good amount of characters for an invertebrate their size. We have about 60 morphological characters across the 27 taxa in our matrix. So yeah, they look like this. They don't look like a whole lot and they are teeny tiny. They're a couple of millimeters long. I don't even know how you find that in your sample. It's amazing to me that we're able to do this. And so I'm going to show you a figure here where in this first graph in A, what we have is basically the standing disparity. So this is a pairwise disparity calculated using the morphological character matrix between taxa. So the blue dots are the kind of cumulative disparity through all time intervals to that point. The X's with the lines indicate standing disparity at that geological stage. What you can see down here, I'm not quite sure why my axis got a little bit funky there, but there are a total of 5 geological stages that we looked at. These are all, 5 geological stages to the late Cambrian period. All right. In B, we have the standing sampled richness plotted against the rescaled disparity. I find this graphic not as informative as this third one, which is kind of the cumulative richness over evolutionary time. Those circles are labeled with the time period that they are from. And what I want you to take away from that is that between this first time period, Woluwian 2 to Woluwian 1, we gain a lot of species richness very quickly in this system. So this follows a pattern that we see in a lot of groups where you have a lot of diversification fairly early in the group and fairly quickly. So from the first appearance to a couple million years into their geologic range, we have a lot of diversification accumulating. Sometimes this is referred to as like an early burst. And so this gave us some idea of what we should look at for our clock models, right? So our clock models, again, describe the way the rate of evolution is distributed through time, right? And so knowing that we have an early accumulation of diversification, we decided, okay, well, we'll have a look at a strict clock, right? Which says, evolutionary rates are evenly distributed through time. They don't really change. We'll have a look at that as sort of a null model. We'll take a look at an autocorrelated clock, which says that the rate of evolution of a descendant should be similar to its ancestors. We'll take a look at an early burst clock, which says we have high evolutionary rates early and decaying through time, and an uncorrelated clock, which says, well, maybe you would think that an ancestor would be close in rate to its descendant, but we're going to let whatever the data says happen, right? We can have wild jumps between an ancestor and a descendant's rate of evolution. So using this knowledge that we have early accumulation of lineages, we picked some candidate clocks, right? There are thousands of evolutionary clocks that you can try to fit to data. we narrowed it down to 4. And so the next thing that we had to look at, if you're not familiar with this sort of graphic, this is from the Geological Society of America. This shows the different geological eons down through stages being sort of the smallest geological unit of measurement. So we had rapid accumulation of lineages during this stage and slower accumulation of lineages during these 2 stages. Right. So therefore, right, I think this makes sense. We might expect those evolutionary rates to change over time, and that is why we picked those 4 models. Something else that's a possibility is that we could have the process of diversification. So the process that is generating our tree, that could change over time too. And so these figures, these were generated using John Alroy's 3-timer methods. And what these are is basically a sliding window approach to have a look at turnover, or the ratio of extinction to speciation over time. And so we looked at the ratio of extinction to origination or speciation for the Cambrian period writ large and for echinoderms as a group. So we estimated a range of sort of possible extinction and origination values that would be plausible given other either invertebrates or given the Cambrian period at large. And this allowed us to have some idea of speciation and extinction rates. And if you're interested in this, Pete Wagner has done a really nice job of putting together a workshop. It's called PaleoDB for RevBase. And this, what he's put together here, is sort of a translation of a lot of classical quantitative paleobiology equations and ideas that you could then use to come up with starting parameters for your model, or for your parameter, to come up with feasible priors, there we go, for your FBD model parameters. Right. And so using this information, we were able to place informative priors on our extinction rate and our speciation rate. Okay. One other concept that I'm going to introduce here is the idea of a skyline model. Right, so these are models that allow our tree to have a different process of diversification, so a different tree model over defined geological periods or epochs or stages in an organism's history. And so we decided to test a few different skylines, one in which each stage, that's the 3 interval, has its own fossilized birth death model, one in which the Wallooian is in its own model and the other 2 are grouped together, and one in which each stage is broken in half. We have some environmental reasons to believe that they might have slightly different sampling probabilities between half stages. So we're not totally out to lunch on 2 of these models. We have good reasons from quantitative biology or paleobiology to think that we have some informative priors. This last model here, the substitution model, this is where the bulk of my actual work in my lab lies. How do we describe the ways in which characters change, phylogenetic characters change over time? Right? A lot of the time, morphology is incorporated by what we call the MK model. This was published by Paul Lewis back in 2001. It's a generalization of the Jukes-Cantor model. And so this makes assumptions similar to the Jukes-Cantor model, like if you have a binary character, 0s and 1s, that you're equally likely to go from a 0 to a 1 as a 1 to a 0. Right? And we can generalize that up to a multi-state character. Right? If you're from the molecular world and you're thinking about, well, what about the morphological equivalent of the GTR model? We do have that. It's that 3rd model down where we have each character state having its own frequency. That allows us to have asymmetrical transition rates. I've also written that out as like a parsimony step matrix, if that's something you're more familiar with looking at. And this is something that Walker Pett and I implemented in RevBase a few years ago, the idea that you might have different frequencies of different characters, and that can allow you to functionally have asymmetrical transition rates. Biologically, there is some hypothesis, and this has been debated in the literature, if feeding and non-feeding characters in synctons evolve at different rates. And so we tested having the characters be unpartitioned, so no partitioning between feeding and non-feeding characters, and no gamma distributed rate heterogeneity. That means that every character in the matrix is assumed to evolve at the exact same rate. We also tested a model in which we had the data were unpartitioned, but we did allow gamma distributed rate heterogeneity. And then we tested a model in which we had the feeding and the non-feeding characters partitioned separately. Right, so we reduced this set of models from kind of infinite, whatever you can dream up for your FBD process, to 10 models that had some amount of credible evidence for them. Right, we kept our data set constant, so we used molecular or morphological characters from a paper by Sam Zamora in 2013. He did a lot of the primary character coding on the synctons, and we got fossil age data from the paleobiology database. We did all of our phylogenetic analyses in RevBase. That is our RevBase logo right there. So this is a Bayesian phylogenetic software. We ran each model to convergence to get a sense for how long that would take, and then we used that convergence value to set up stepping stone analyses. Stepping stone analyses are important because they allow you to calculate a precise model likelihood, which if you're comparing between different models, you need to know exactly the likelihood of your model. And so we calculated precise model likelihoods for each of our 10 competed models. And I'm going to show you these here. I've populated the table with different values. We use Bayes factors to compare between different models. That's not terribly exciting to talk about. If you have questions about it, you're certainly welcome to ask me. So for our character model, we found that that partition between feeding and non-feeding characters fit the data the best. On our strict clock, we were sort of surprised by the rejection of the early burst clock, and we found that the uncorrelated clock fit the best. I put a big arrow in here because I wanted to talk a little more about this. We did our early burst clock with sort of high rates of evolution early on and decaying towards the group's extinction. There were other ways that we could write out that clock potentially. The uncorrelated clock is the most parameter rich. And so to me, I sort of look at this result and I'm saying, did we reject this model because there is a different formulation of it that might be better? And so we rejected it in favor of the most, loosey-goosey parameter rich model. So that's something that I think I'm probably going to revisit sometime in the future. Right, so just to show you what these different models look like, up top is that uncorrelated clock, and I've drawn in a sample tree there where we have the evolutionary rates represented with kind of cool colors being low evolutionary rate, warm colors being high evolutionary rate. And what you can see on that tree is that we can have large jumps from, a cool colored low rate ancestor to a high colored or hot colored high rate descendant, and then back to a low evolutionary rate descendant of that. So the uncorrelated clock is extremely flexible. So that could indicate that, truly this is the best model for it. It could indicate that, maybe we're just modeling something less than adequately using that early burst. So I haven't fully given up on that. And then for our tree model, what we found is that each geological stage having its own tree model was the best fit for this data. And I was a little bit comforted by this because this is sort of the medium complexity model. We didn't just go straight to the most complex thing. So to me, that says that we're probably capturing some true signal. All right. So again, I wanted to note that, we didn't evaluate every possible fossilized birth death model, right? We could have split this into, each million-year period has its own FBD model. We didn't do that. We used our knowledge about quantitative paleobiology, about the geological record in this group, to choose a few models to evaluate. And so to me, something that is sort of important is that we're making a little bit of a return to hypothesis-driven work, right? We have a hypothesis that feeding and non-feeding characters might evolve differently, and we are using that to narrow down the range of character models that we're going to look at. And so we're not just throwing all of our data and making the most complex model we can. We're doing a careful examination of what we think really is the model. And so what was the result of this? Not a whole lot that's incredibly novel. So you can see in blue there, that's what we call the Suchocystidae. That's pretty much always supported as a monophyletic group, and we find it as almost a monophyletic group here. Same with the Gyrocystidae. The Trochocystidae is often sort of paraphyletic, and that's true here too. We did find support for a number of sampled ancestors. So I've indicated here where you see the name of a taxon with an arrow drawn to the name of another one. That is, the first name will be the ancestor, the second name will be the descendant. So we did find a number of sampled ancestors, and we were a little bit surprised by that. Because this is a fairly low completeness record, we think we probably have sampled about 21% of the diversity in this group. And so we can actually use some calculations made by Mike Foote in the 70s to figure out exactly how many sampled ancestors we should have. And it should be about half as many as we do have. But syncytins are also really geographically restricted, which might increase our probability of finding sampled ancestors. Something that was sort of interesting is this Protosynctus down here. The placement of this has been kind of controversial. As the name suggests, proto first, right? For a long time, people thought that this was the earliest diverging member of this group. Parsimony analyses have put it in sort of various places, including up in that Suchocystidae. That's why it's in blue here. And we return it back to sort of that earlier divergence in the group. Now, something that I think is really exciting, and I'm just going to tease this a little bit, we don't treat the fossil ages in this analysis as completely known, right? So we treat the age of the tip as something that you can sample, right? So here I've indicated we have an observed minimum and observed maximum. That is how young, like what is the oldest age you think this fossil could have? What is the youngest age you think this fossil can have? And so we parameterize this with a uniform distribution between the two. And what we found in this group is that for some of those tips, we're able to actually not just get the prior back. So that very first one, Suchocystis acorophora, that was a uniform prior between 3 and 5 million years. It looks like this fossil might come from, so that should be 500 to 505 million years. This fossil looks like it might come from the younger range of that group. So this is something that had been suggested as an application of the fossilized birth death. Perhaps in high uncertainty fossils, we can narrow that range. And here we actually did find some evidence for being able to do that. So that's kind of exciting. So for this part of the talk, I'm just going to wrap up with some lessons. There are many ways to write out a fossilized birth death model, right? There are many ways to say, oh, well, I think this varies over time, but this does not. There are many ways to write out variation in the subcomponent models. You can test whatever character evolution models you want. Pretty much infinite, right? But in practice, you will waste years and years of CPU cycles trying to test all the models. And so I think what we did here, and we did a nice job of, is kind of narrowing down the possible values and possible models that we need to test. And I think that's going to be something that's important going forward because even though we only tested 10 models, that was a fairly time and compute intensive process. So we can't possibly do it all. And I think we've done a nice job of showing some ways that you can narrow some things down. And in my last few minutes, I'm going to talk to you about a data set that's sort of on the opposite end of the scale from syncytins. So the syncytin data set is nice. It is well behaved. It's small, but it's fairly complete. There's not a ton of missing data. It's temporally restricted. Their synctons are only found in about a 10 million year period. There's fairly narrow ages or uncertainties on ages. Something that I've been working on is doing fossilized birth death dating with ants. So this group probably isn't older than 200, oops, sorry, let's say MY. It's probably not older than 200 million years old. They're found pretty much everywhere, which can make it hard to sense how complete our sampling is, both in the present and the past. There are many, many species, but also there are known biases in how we collect data. And so this, for example, these are some ants that are preserved in amber. You can see you can get beautiful morphological characters from there because they are very beautifully preserved, right down to the behavior, right? Those ants are having an aggressive interaction. One is biting the other one on its face, right? So this is an ant bar fight that has been preserved for eternity. Our fossil samples are often, even though individual fossils may have yielded beautiful morphological data, they are taxonomically biased. So here, what I have on the x-axis is the subfamily of ants that a fossil belongs to and how many fossils there are. We can see our Formicidæ, our Dolichoderinae, Ponerainae, Myrmicinae, tons of fossils. This looks like we have a skyline where we have skyscrapers and tumble down shacks, right? There are some groups that are very depauperate in fossil samples. Fossil samples are also sort of temporally biased. I'm pretty sure ants didn't go extinct at like 65 million years and re-evolved at 55 million years, right? We just don't have good sampling in that exact period. So we know that we have some temporal biases. We know that we have some taxonomic biases. And that's also true of the molecular data. We have some known biases like the side of the road bias, where when things are easy to collect, we tend to collect them more. Things that are from near human habitation are often better characterized and have more data. And there's been some systematic conflict in this group. So this is a figure that was adapted from a paper by your colleague, Marek Borovic, where he outlines several of the common molecular phylogenetic hypotheses. Morphological data often sort of don't really agree with any of them unless you use clade constraints. And there's also some kind of age conflict, right? Morphological analyses often support a younger age, molecular analyses often support an older one. And of course, all of that tends to depend on what exact assumptions you make in your analysis about the way in which your character data evolved. And so I got to wondering, can we resolve some of this conflict by accounting for how fossils are sampled? Right, so we did some initial explorations, and this is part of the talk where I'm going to talk about, ongoing work. So I don't have a whole lot of results here. I just have a head full of questions about what's rapidly becoming my white whale. What we have here is, initially we tried estimating kind of a time homogeneous fossilized birth death model. Right? So we have one rate of speciation that applies to the whole tree, one rate of extinction that applies to the whole tree, one rate of fossil sampling that applies to the whole tree. And based on what I just showed you, we would expect that this doesn't fit very well. That is precisely what we find. My master's student, former master's student, she's now a PhD student at Oklahoma State, Christina Kolbman, did some posterior predictive model adequacy testing. And these are simulation-based tests where you take output from your Bayesian analyses and you use that to seed simulations of whatever you're trying to simulate. In this case, we took values for speciation, extinction, and sampling from our posterior trace, and we tried simulating trees under the fossilized birth death, starting from those parameters. And what we discovered, we did this 3 different ways. That's what these 3 different colors indicate. What we discovered is that the root age, or the origin time, for the trees that we simulate with these time homogeneous values are much younger than is plausible. no other phylogenetic analyses have concluded that ants are 48 million years old. And what that tells us is a model in which we have one speciation rate, one extinction rate over the whole tree is not adequate for these data. So I'm gonna black bar that hypothesis out. We also tried one where we have kind of different sampling regimes over the tree. So for example, in this period where we have 75 to 100 million years, perhaps there's one fossilized birth death process in that one. Perhaps there's one in this second interval, and perhaps there's one kind of describing 25-ish million years to the present, right? So each interval gets its own FBD process, right? We tested one other or two other models. One is when we give each interval its own fossil sampling value. Right, so we're not assuming that speciation or extinction change over time, but the rate at which we are sampling fossils is what's changing. Okay. We also tried linking these across this. So perhaps if you have high fossilization in one interval, that's because there is high fossilization in the previous interval. This had a terrible, terrible model likelihood. So we're sort of zeroing in on different scenarios in which we have either the FBD writ large or only fossil sampling varying over the tree and over time. Right. So this is an area of active research where we're trying different sets of kind of Skyline models for the fossilized birth death or for just the fossil sampling parameter of that. My personal inclination, and this is just between me and however many of you there are, I think what's actually changing across the tree is the fossil sampling rate. But talk to me again in like 2 years when I've actually done all of the experiments, okay? And so something that we have detected so far is that these are initial posterior traces, is that it does appear, so on the left-hand side, we have the fossil sampling rate. If we look at sort of chunking up ants' evolutionary history into 3 intervals, what we do see is that we do have different rates of fossil sampling across those intervals. That's not as true with the speciation rate, where it looks like there might be a little bit of peakedness to one of them, but it looks like there's not a whole lot of difference in the speciation rate over time. So I think probably we have fossil sampling varying throughout time, but this is work that we're really just ramping up in the lab. So this is TBD. Now, something that's interesting about this is that we do get radically different ages for the fossils. So sorry, that should be time homogeneous. When we estimate it, we get a little bit of an older age than we did in our simulations. I'm not quite sure what's up with that. When fossilization varies over time, we get ants being about 140 million years old. We tend to get about the same topology between all of these models. It's just the age that's varying. When we have all of the FBD parameters varying over time, we end up getting an age of ants that's about 190 million years old, which is very old relative to other literature estimates for this group. And so we're, like I said, we're just starting this journey with doing these more exciting model fittings. So what we're looking forward to doing in the lab right now is expanded posterior predictive model adequacy testing. So doing some more advanced simulations where we incorporate more information about kind of skyline modeling or skyline dynamics. We're looking at doing some estimation of kind of clade-wise biases. So if we don't have the full FBD varying through time, are there individual branches that might be better fit by having their own model? So we're going to be taking a look at this. And something that kind of sits poorly with me is when we have different model components, right? So for example, our clock and our tree model, if we did have an early burst clock, that's not wholly independent of the tree model, right? So when we have interactions between different parts of the model, how well are our model adequacy and model fitting tools working in those particular situations? Something else that we're working on is those of you who have used RevBayes, which is where we did all of our phylogenetic estimation. Those of you who have used RevBayes might have observed that it can be a little bit clunky to work with, especially if you're doing visualization. So we've been working on this package, RevR, which allows you to use RevBayes in the RStudio window, including with knitr. And so you can take advantage of some of RStudio's graphical interfaces. So for example, this is a plot from a paper that I wrote with Graham Lloyd this past year. If you wanted to do something like a tree space visualization and look at the way your MCMC is exploring tree space, ideally what you want to be able to do is, go from RevBayes with your sample of trees and just import that straight into R and do your visualization right at the end of your MCMC run. And so that's something that we're working on, kind of leveraging some of the advanced graphical capabilities of R to make it easier to look at posterior samples. Finally, swear I'm at the end, although I'm doing fine. Okay. I am hiring a postdoc. On my website there, I have kind of a description of the job. The job ad should be live soon. But this is a teaching and research postdoc position on my career award. So there's kind of some educational aims that you would be taking part of if you wanted to apply for this particular position. To be doing fossilized birth death stuff in the lab and a little bit of teaching. Just wanted, since I had a captive audience, wanted to put up a postdoc ad. And with that, I want to thank everybody who made this visit possible. This has been a really wonderful day. I want to thank the members of my lab, Basanta Cockerell, Caleb Charpentier, Shreya Bhatt, Courtney Grigsby, Tyler Tran, and my former master's student, Christina Coleman, with a bunch of collaborators here. I've included a link to that postdoc job and then, of course, my funding sources. And I believe I have time for questions.
Thanks, April.
Should I ask one?
Go for it.
This is stepping back a bit. So I haven't kept up with this part of the field, but from time to time, people encounter data that are incongruent with other data. You build a tree, and then if you were to go back and look, some of the data wouldn't, they just absolutely be statistically incongruent. Do you have a sense of how incongruence increases or decreases as you go to these increasingly parameterized models, the models with more processes and so on? I mean, it could go either way, right? Because one way to get rid of incongruence is to add processes to the model of evolution. I just don't have a sense of this.
I don't have a sense of it either. And so I think in this particular data set, with the depth of divergence, we have some kind of saturation and sorts of issues like that. I also think with ants in particular, like there are only so many ways to make an ant, right? And so we have a pretty high convergence morphological data set. And so like, there are some issues with the substitution process that make resolving this phylogeny fairly tricky. There are also fairly deep in the tree, some very long branches. And so I don't necessarily even know if incongruence is our issue as much as it is just these overlapping and extremely systemic biases to different parts of the data. And I mean, it is possible that like one data set is just lying to us, right? And it's probably the morphology if that's true. But so yeah, so I think incongruence is a concern, and I think, I don't know, just in terms of approaching it, we're gonna have to really get nitty and gritty with this particular data set.
I have kind of like a naturalist kind of question about your ant work. Is there any support for different rates of fossilization from the tree paleontological work? Like are there? Yeah, exactly.
Yeah, so something that I need to go through and do is sort of, actually, no, I'm hiring. What I need to have my postdoc go through and do, there we go. Sorry, still getting used to that. Is some of those amber deposits have extremely wide kind of confidence intervals on how old we think they are. So I think something that we could do is kind of doing an FBD process where the probability of fossilization is directly tied to whether or not we have one of those deposits kind of forming during that time. So those sorts of really biological ideas. And that's something that, like, certainly I'm pretty excited to look at, because I think, like I said, you know, between me and my, what are we, 35 closest friends here, I do think that what is probably actually varying is maybe not the speciation rate, but it might be the fossil preservation rate.
Okay, you mentioned that having an ancestor and an extant species sampled on the same branch, like maybe vertebrate people were less comfortable than invert people. Yeah. Like, what's that about?
That's just an observation. Let me find the slide where we talk about that. Where are you? There we are. Right, so in that bottom panel where we have basically a 2-degree node, so what you have there is, you know, some population of organisms, you know, they continued existing, right? They didn't, kind of the traditional, particularly vertebrate paleo mode of thinking is, if you have something sampled, it's because it went extinct at that point, right? So if something has gone extinct, then it's not leaving any descendants on the tree. But that way of thinking about things is less common in the invertebrate paleontological world. And I'm not quite sure what explains exactly that difference. But they seem much more accepting of the idea that you might have a population that leaves descendants and it's sort of anagenic change and then speciation somewhere down the line. And so particularly Mike Foote with brachiopods did a lot of the pioneering work on modeling how and when you expect to see ancestors have sampled descendants in the fossil record. And so I was working with Pete Wagner on this paper, and he's sort of Mr. Quantitative Paleobiology. And I was like, that seems like a lot of sampled ancestors. And he goes, hold on. And he puts me on mute and then calculates the probability that we should have more than one sampled ancestor. And he goes, it's a little high, but they're also geographically restricted. So I think that's okay. I kind of went, oh, okay. So you've been thinking about this since like 1990. Okay, that's fine. So it seems like in the invert world, they just think about that a little bit differently. And when you look at kind of the quantitative paleobiological literature, a lot of it is in either marine or freshwater invertebrates.